Imagine a construction crew, right?
Right.
And they decide to build this massive sprawling bridge.
Okay.
But instead of using steel or concrete, they construct it using the very cars and trucks that are driving underneath it.
Oh, wow.
Yeah. And if the traffic is flowing, the bridge holds strong. But the second you stop that traffic, the entire structure just completely vanishes into thin air.
It sounds like a physics-defying magic trick, but it's actually one of the most insidious survival mechanisms we've ever found inside a human cell.
Welcome to your custom deep dive. Today we're taking a real magnifying glass to a 2026 paper from the journal Molecular Cell. It's published by Budenich and this brilliant team of researchers.
A fantastic paper.
It really is. And the mission today is to understand how certain super aggressive childhood cancers basically pull off exactly that magic trick. They literally hijack the local traffic in our cells to build toxic tumor-driving factories.
And we should probably establish the clinical stakes right up front because they are incredibly high here.
Okay.
We're primarily talking about pediatric acute myeloid leukemia, so AML and Wilms tumor.
Which is a childhood kidney cancer, right?
Exactly, a very aggressive childhood kidney cancer. And in these specific diseases, the culprit is this single protein called ENL.
Okay, so let's unpack this, because normally ENL is one of the good guys, right? It's a helpful protein.
Yes, normally it's essential.
But then it undergoes this tiny mutation that turns it from a helpful cellular worker into an absolute monster. So before we talk about the mutation, what exactly is wild-type ENL? Like, what is its actual day job in a healthy cell?
Let's set the baseline. So in a healthy cell, wild-type ENL is what we call a chromatin reader.
So chromatin reader.
Right. Now, to understand that, you kind of have to visualize how your DNA is stored. You have about 6 feet of DNA crammed into the microscopic nucleus of every single cell.
Which is still hard for me to wrap my head around.
It's a lot of material. So it doesn't just float around loosely. It's tightly wound around these little protein spools called histones.
Like thread on a spool.
Exactly. And that whole complex, the DNA plus the protein spools, that's called chromatin.
So it's essentially this massive coiled up library of instruction manuals.
Yes.
But, I mean, you can't read a book if it's tightly packed away in a moving box.
Precisely. The cell needs a way to flag which boxes to open and read. So it attaches these tiny chemical tags, specifically acetyl groups.
Acetyl groups.
Right. It puts them onto those histone spools. It's basically a chemical green light. And wild-type ENL has a highly specialized structural region called a YEATS domain.
YEATS.
Yeah. Think of the YEATS domain as like a barcode scanner. It scans the chromatin, reads that acetylated green light, and physically binds to it.
Oh, so it grabs onto the green light.
Exactly. And once it's attached, ENL recruits other proteins to help activate normal gene transcription. It literally just helps turn the right genes on at the right time.
So it's like a polite, highly specialized office worker doing a specific job at a specific desk.
That's a perfect way to look at it.
But in pediatric AML and Wilms tumor, this ENL protein suffers from what you call a gain-of-function mutation. The paper highlights these tiny errors called T1 insertions or T2 deletions.
Right. And what that means is a minuscule string of amino acids is either accidentally added or removed right inside that YEATS domain barcode scanner.
It's just a microscopic typo.
A tiny typo, but the consequences are huge.
Which brings us to the core mystery of this whole deep dive. How does a typo that small change the physical behavior of this protein so drastically that it triggers a fatal childhood cancer?
Well, to understand that, we actually have to look at the physics of the cell nucleus, specifically a concept called liquid phase separation.
Phase separation. Let's break that down. I always picture, shaking up a bottle of vinaigrette dressing.
Oh, yeah, the oil and vinegar.
Right. You see those tiny droplets of oil completely separating from the vinegar and clumping together into these little floating orbs.
That's a really great visual. And inside the chaotic, watery environment of the nucleus, proteins can do the exact same thing. They condense into these high-concentration liquid droplets. We call them biomolecular condensates.
Biomolecular condensates, got it.
Yeah. And when ENL gets that T1 or T2 mutation, it fundamentally alters the protein's physical properties. It suddenly becomes incredibly sticky to itself.
Oh, so they clump together.
Way too well. The mutant ENL proteins self-associate and form these aberrant, toxic, oil-like droplets at very specific DNA targets, particularly at known cancer-linked genes like the HOXA gene cluster or CBX3.
Okay, so returning to our analogy, wild-type ENL is the polite office worker, but mutant ENL is like a rowdy flash mob.
A very sticky flash mob.
Because they're so sticky, they congregate at a very specific street corner, let's say the HOXA gene, and they form this massive crowd that just refuses to leave.
It's a localized traffic jam, but the real danger is what they're actually doing while they're crowded there.
What are they doing?
Well, because these mutant condensates are sitting right on the gene's promoter, which is the on switch, they cause what researchers call frequent transcriptional bursting.
Oh, so they're basically shouting into megaphones.
Essentially, yes. They are holding that on switch down permanently, and this leads to hyperactive transcription, which is a massive overproduction of RNA from those specific genes.
And that's bad because
that flood of RNA is exactly what instructs the cell to abandon its normal duties and become a rapidly dividing cancer cell.
Okay, but I have to push back a little on this flash mob idea.
Go for it.
Because, think about your own physical space.
Yeah.
What happens when too many people crowd into a tiny room? It gets chaotic. And you say the nucleus is chaotic. It's full of molecular machinery flying around at incredible speed.
It is very violently active, yeah.
So just being slightly sticky from a tiny genetic insertion doesn't seem like it should be enough to hold a massive liquid droplet together against all those physical forces. Shouldn't the crowd just get ripped apart?
And that is the exact paradox the researchers noticed. The mutation initiates the gathering, sure, but it really shouldn't be enough to sustain a massive, stable droplet on its own.
So they needed help.
Exactly. They hypothesized there had to be an accomplice, something in the local environment acting as the mortar between the bricks, keeping them glued together.
And the accomplice they found is RNA itself, which is just wild to me because we usually think of RNA as just a passive messenger, right? Like it's just the disposable blueprint the cell uses to make proteins.
Usually, yes.
But here, the researchers are saying RNA is acting as a physical building block.
Yeah.
So how did they actually prove that ENL physically binds to RNA? Because seeing an interaction that microscopic has to require some serious biochemical detective work.
It does. And they started with a classic in vitro technique called an EMSA. That stands for electrophoretic mobility shift assay.
EMSA. I've always thought of EMSAs as a sort of microscopic obstacle course.
That's functionally what it is. You take a slab of porous gel, put your molecules at the starting line at the top, and you apply an electrical current.
Okay.
The gel acts like a sieve. Small, unattached molecules race through the tiny pores really quickly and end up at the bottom of the gel. But if a protein physically binds to a piece of RNA, they form this heavy, bulky complex.
So they get stuck.
Right. When you force them to run the race, that bulky complex gets tangled in the pores and gets stuck near the top. Its mobility has, quote-unquote, shifted.
Oh. So they purified ENL, mixed it with a piece of single-stranded RNA from the HOXA11 gene, and they ran the race.
Exactly. And the result, both the wild type and the mutant ENL naturally bound to the RNA. They formed that bulky complex and got stuck at the top of the gel.
So they proved the physical connection.
They did. But proving that they bind isn't enough. You have to know how they bind. Like where on this entire three-dimensional protein is the actual grabber?
And that's a much harder question.
A lot harder. So for that, they utilized AlphaFold 3.
Oh, the AI system. The one that predicts 3D molecular structures.
Yeah. Yes, and it is remarkably precise. They fed the sequences into AlphaFold 3, and the AI pointed to a very specific spot inside that YEATS domain barcode scanner. It highlighted what we call a basic patch.
A basic patch.
Yeah. It consists of specific amino acids, lysines at positions 61 and 67, and arginines at positions 63 and 64.
Wait, hold on. Let me dust off my high school chemistry for a second here.
Sure.
The patch is basic. That doesn't mean simple. It means it has a positive electrical charge, right? Because lysine and arginine are positively charged amino acids.
You've got it perfectly.
Yeah.
Now, think about the RNA molecule. RNA has a sugar phosphate backbone, and phosphates are heavily negatively charged.
Opposites attract. The positive basic patch on the protein acts like a magnet for the negative backbone of the RNA.
Exactly.
It's literally molecular Velcro. The positive charge is the lysines and arginines are the little microscopic hooks.
Yeah.
And the negative RNA is the fuzzy fabric.
It is exactly like Velcro. And to test this Velcro model, the researchers did something brilliantly elegant. They engineered a brand new version of the mutant protein.
What did they do?
They took the cancer-causing T1 mutant, but they went in and surgically swapped out 3 of those positively charged amino acids, the lysines and the arginine, and they replaced them with an amino acid called alanine.
Okay.
They named this new engineered version the T13A mutant. The 3A stands for the 3 alanines.
Why alanine specifically? Like, why not something else?
Because alanine has a very small, non-reactive, electrically neutral side chain. It doesn't have a positive or a negative charge.
Oh, I see. So they chemically shave the hooks off the Velcro. The 3A mutation basically turns the sticky Velcro into just smooth tape.
Exactly. But here is why that is such a powerful scientific control. Swapping those 3 amino acids to alanine didn't break the rest of the protein.
It didn't.
No. The AlphaFold models in the physical tests confirm that the ENL protein maintained its exact 3D shape, and its YEATS domain could still bind to the acetylated DNA perfectly fine.
Wow.
The only thing they destroyed was its ability to grab RNA.
Okay, let's pause and make sure we have this straight. I want to give the listener a quick recap because this is the pivot point of the whole paper.
Good idea.
So, bullet 1. We know mutant ENL proteins get sticky and form these toxic oil droplets on genes, causing them to burst with overactivity. Bullet 2. We know RNA is acting as the structural glue holding these droplets together, attaching via a positively charged patch. And bullet 3. Scientists have built a 3A version of the mutant protein that is identical in every way, except it is completely blind to RNA.
They created the ultimate tool to test what happens when you pull the structural rug out from under the tumor.
Which takes us out of the computer models and into real life. They started with the in vitro droplet assay. So they took purified mutant ENL, put it in a tube, and it formed little microscopic droplets. But when they added the HOXA11 RNA, boom, the droplets massively expanded.
The RNA supercharged the phase separation and made the droplets huge. But when they ran the exact same test, using the T13A mutant, the one with the shaved-off Velcro, it completely ignored the RNA. The droplets stayed small and weak.
But obviously, human cells are a lot more complicated than a purified protein in a test tube. How did they prove this exact interaction was happening live inside a living cell?
Right. In vivo is always the harder proof, so they used a technique called CL-EPSIC.
KLP, yeah.
It stands for cross-linking and affinity purification followed by sequencing. Basically, you take living cells and you blast them with a very specific wavelength of UV light.
Wait, UV light, doesn't that just fry the cell?
It's a very fast, very controlled pulse, and what it does is instantly form permanent covalent bonds between any protein and any RNA molecule that are physically touching each other at that exact millisecond.
Oh, so it freezes the interaction in time.
Exactly. It glues them together permanently. Then you extract the ENL protein under incredibly harsh chemical conditions.
Meaning you aggressively wash away anything that wasn't permanently glued to ENL by that UV flash.
Right. So if an RNA survives that harsh wash, it absolutely had to be directly touching the ENL basic patch when the light flashed. And when they sequenced what was left, they found that ENL was heavily bound to unspliced pre-mRNA.
Pre-mRNA. Okay, that prefix is crucial. That means ENL isn't just floating around the cellular cytoplasm catching mature RNA that's drifting by. It is grabbing the RNA right as it is literally being born off the DNA template.
It's catching it fresh off the press. The ENL is sitting on the gene, forcing the gene to make RNA, and then immediately using that freshly minted RNA to build a larger droplet to force more transcription.
It's a toxic feedback loop.
A massive one.
Which brings us back to that bridge made of traffic analogy from the very start of the deep dive. The researchers wanted to know what happens if you disrupt that local traffic. So they ran a displacement experiment using a chemical inhibitor called TDI-110155.
Right, so TDI-110155 is a drug that competitively blocks the YEATS domain.
Competitively blocks. So it competes for the same spot.
Exactly. It basically wedges itself into the barcode scanner, so ENL can't hold on to the chromatin anymore. It physically forces the condensates to disperse off the DNA.
It breaks up the flash mob.
Exactly. They applied the drug, the droplets dissolved, but then they washed the drug away to see how well the condensates would reform a process we call renucleation.
Right. Will they come back?
And in cells with the standard cancer-causing T1 mutant, the condensates rushed back to the DNA and completely rebuilt themselves within 10 hours.
But let me guess. In the cells with the T13A mutant, the one with no Velcro, they couldn't do it?
They severely struggled to reform. Without the ability to grab the local RNA, they just couldn't rebuild the flash mob.
And this led the researchers to a brilliant follow-up question. If the RNA is the scaffolding, do we need new scaffolding being built constantly?
Right. Is old RNA enough, or does it have to be active transcription?
Exactly. So to test this, they bypassed the ENL protein entirely, and they targeted the RNA factory itself. They used transcription inhibitors, drugs like actinomycin D.
Which blocks transcription from even starting. Yeah.
and DRB.
Which stops it once it's already going.
So they essentially halted the traffic.
And the result was stunning. Blocking new RNA synthesis perfectly mimicked the 3A mutation.
Wow.
Even with standard, fully functional mutant ENL proteins, if you stop fresh RNA from being made locally, the toxic condensates simply fall apart. They cannot rebuild.
So let's do another quick synthesis here for you listening. The RNA isn't just an accomplice, it's a structural requirement. Locally produced RNA acts as a real-time scaffold. Without active transcription, or without the protein's ability to magnetically grab that RNA, the cancer-causing condensates literally cannot sustain themselves. The bridge vanishes.
Which is just an incredible mechanism to uncover. But up to this point, we've only been talking about cells in a dish.
Right.
The ultimate test of any cancer biology is whether it holds true in living physiology. Does breaking this RNA Velcro actually stop the disease in a living body?
So it's time to look at the mouse models. But before we get to the actual mice, we have to talk about where the mutant ENL actually sets up shop. Because it doesn't just form flash mobs randomly everywhere, right?
No, it has very specific tastes. The researchers used a sequencing algorithm called chromatin immunoprecipitation sequencing.
Okay, chippy-sec.
And they used it to map exactly where mutant ENL goes across the entire genome. They found that it scatters everywhere. It binds to thousands of different sites. They call these low-bound sites.
But it's not causing cancer everywhere.
Correct. It only forms those massive, hyperactive droplet condensates at less than 1% of its targets. These are the top-bound or permissive sites, like the HOXA cluster we mentioned earlier.
Right.
And the data revealed a critical distinction. RNA binding is heavily, preferentially required at these top-bound sites.
I picture it like the difference between two people waiting at a quiet bus stop that's a low-bound site versus a massive riot outside a stadium, which is the top-bound site.
Okay, I like that.
Because a couple people at a bus stop don't need megaphones and organizational scaffolding to just stay there. But the riot at the stadium, if you take away their communication and their scaffolding, the massive crowd falls apart.
That's a really great way to visualize it. The 3A mutation removing the Velcro barely affected the low-bound sites. But at the top-bound sites, ENL occupancy just plummeted. The massive condensates collapsed.
Oh, wow.
So, armed with this genomic map, they finally moved to the transplantation model in mice.
Walk us through the setup, because how do you give a mouse this specific type of leukemia?
You start with healthy blood stem cells from donor mice. Specifically, we call them LSK cells.
LSK cells.
Yes. You divide them into 3 groups, and you use genetic engineering to insert 3 different versions of the ENL gene into them. Group 1 gets wild-type ENL, Group 2 gets the cancer-causing ENLT1 mutant, and Group 3 gets our engineered ENLT1-3A mutant, the one with the shaved Velcro.
So all the stem cells are exactly identical, except for exactly which version of ENL they are forced to express.
Precisely. Then you transplant these stem cells into recipient mice, but there's a catch here. You have to lethally irradiate the recipient mice first.
Because you need to completely wipe out their existing immune system. You need them to be a blank slate so they entirely adopt the transplanted blood cells as their own.
Yes, otherwise they'd just reject the cells. So the mice begin pumping blood generated by these new stem cells, and almost immediately, they could see a difference at the molecular level.
That fast?
Yes. They use a technique called GSEA, gene set enrichment analysis, to look at the gene signatures. And the mice that received the T1 mutation lit up with aggressive leukemic stem cell signatures. The cancer factory had officially opened for business.
Now for the physical results. The mice transplanted with the standard ENLT1 mutant developed aggressive, massive tumors.
Right.
They suffered from profound splenomegaly, meaning their spleens became incredibly enlarged as the cancer took over their blood filtration. Leukemic blasts infiltrated their lungs, their livers, their kidneys. They died rapidly. It's a brutal, fast-moving disease.
It is, but the third group is where this paper makes history. The mice transplanted with the T1-3A mutant.
Right.
Remember, this is the exact same cancer-causing T1 insertion.
Right.
The protein still has the mutation that makes it sticky. The only difference is that 3 tiny, positively charged amino acids were neutralized.
They just turned off the Velcro.
Those mice had 100% survival after a full year.
100%.
No leukemia at all. Their spleens were a normal size. Their organs were completely clear. By merely preventing the mutant protein from grabbing the local RNA, they completely abolished the leukemogenesis. The cancer couldn't even get started.
That is just jaw-dropping. It didn't just slow the cancer down or make the tumors smaller. It completely prevented the disease from taking hold in the body.
And this leads the authors to establish what they call a threshold model. And it fundamentally changes how we view these mutations.
How?
Because the T1 mutation alone isn't enough to cause cancer. Yes, the mutation makes the protein sticky, and yes, it starts to gather at the HOXA gene. But to cross the dangerous threshold into full-blown, uncontrolled tumorigenesis, it needs that local RNA. The RNA is the scaffolding that pushes the condensate nucleation and the gene bursting over the edge.
Breaking the ENL-RNA interaction locally reduces the condensation and the gene bursting just enough, just slightly below that dangerous threshold, to completely prevent leukemia in a living mammal.
It proves beyond a shadow of a doubt that RNA is a functional required cofactor in this oncogenic process.
Imagine you're holding a heavy weight on a delicate glass table.
Right.
The weight itself might not break the glass, but if you add just a single heavy textbook on top of it, the glass shatters. That textbook is the RNA. If you can chemically remove the book, the table holds.
I love that. And that's where the broader scientific impact of this paper really comes into focus, because this discovery isn't just about ENL or pediatric AML or Wilms tumor.
Right, because there are a lot of cancer-causing proteins out there.
Exactly. The authors point out that other massive, notorious oncogenes like NUP98-HOXA9 and mutant NTM1C, they also have these intrinsically disordered, highly charged regions.
Wait, you're saying they might be using the exact same magic trick?
It is highly likely. They might also secretly rely on freshly minted local RNA to form their own tumor-driving condensates. What Budenich and her team have uncovered here is likely a fundamental biological trick that multiple types of deadly cancers use to survive and thrive.
So if you're listening to this, why should you care about the microscopic basic patch of one specific protein? Because the textbooks on how cancer works are literally being rewritten right now. For decades, biology classes taught us to think of RNA mostly as a passive, disposable messenger tape.
Right.
But now scientists have to include the physical, architectural properties of RNA as a crucial piece of scaffolding in our models of human disease.
We're moving beyond the old model of just trying to drug the mutant protein directly. We are now looking at the environment that allows that mutant protein to function. We're looking at the scaffolding.
Which leaves us with a highly provocative final thought. If local RNA acts as the required structural foundation for cancer-causing proteins, could we design an entirely new class of future drugs?
That's the dream.
Drugs that don't even try to attack the cancer protein, but instead just chemically scramble the local RNA scaffolding. Imagine a therapy that simply dissolves a floor beneath the tumor, and we get to just watch the cancer droplets harmlessly melt away. It completely redefines what a therapeutic target can be.
It's an entirely new avenue for therapeutics, and it gives us a profound new vulnerability to exploit in some of the most stubborn diseases we face.
Think back to the start of this deep dive, the bridge built out of its own traffic. It seemed impossible to dismantle without blowing up the whole road. But today, we've learned that you don't need explosives. You just need to know how to turn off the traffic lights. Until next time, keep diving deep.